Hi everyone, it's Mr. Sinti, and today I have the pleasure to discuss carbohydrates with you. Now, carbohydrates, I think most people are familiar with them, sort of fundamentally, that these are sugars, and they are very sweet, like, for example, found in these figs down below. But what's fascinating about carbohydrates is that they can come in simple sugars, and these simple sugars could be linked together to form larger and larger polymers, and the functionality is quite remarkable. It spans from energy storage all the way to informational molecules. Like, for example, sugars are found in DNA and RNA. And then sugars can also be found on the outside of cell membranes and transport vesicles in terms of identification. So it's a myriad of functionality, and let's get right into that conversation. I think most people love carbohydrates. I'm going to go out there and say that because they're sweet, and I think most people enjoy that. And clearly, they're important sources of energy. And I just want to just start off with that because that's the main function. But they could be simple and they could be really elaborate, like I was saying before about polymers. And so, what we do with a carbohydrate and sugars in general is that we take that prefix that we're familiar with, mono meaning one, and we throw in this suffix called saccharide. And so, this sort of replaces the mer, which is a generic part. So, it's a monosaccharide or a one sugar. Simple sugars are considered to be monosaccharides. And then, logically, when you connect those two together, two monosaccharides, it forms a disaccharide, and those are formed through a condensation reaction or a dehydration synthesis. Those both mean the same, where water is liberated when those two monosaccharides come together to form the disaccharide. And then if you link a few more together, it's called an oligosaccharide. And oligosaccharides are really significant as well. Those are those sugars I was alluding to that are found on the outside of the plasma membrane, which are informational. And then you can get these big polymers, these big polysaccharides to form. And those are quite important for living organisms. Those can be either big energy storage, because you can imagine there's many, many, many sugars involved, uh, or they could be structural, like, for example, found in plant cell walls in the form of cellulose. And so we're going to get into this. We're going to get into what's the story? What are some general characteristics of carbohydrates? We're going to talk about some examples of monosaccharides, di, oligo, and poly, and we'll get into a little bit more detail of functionality. So, altogether, a sweet conversation about carbohydrates. So, one of the first things that I want to say right out of the gate is that sugars are traditionally formed through the process of photosynthesis. So, the sun comes down and it hits any sort of chlorophyll. And in this case, I'm choosing a land plant called the sugarcane. And plants are able through photosynthesis to produce sugar. And this is how they're able to synthesize carbohydrates, through photosynthesis. And so that's how these carbohydrates get into the world, through photosynthesis. And again, it's the green structures in particular, like, for example, sunlight is coming down and it's producing these sugars here. Now, what's interesting is the sugar could either be consumed as energy by the leaf, or it could be shuttled through vascular tissue, shown here, and it could be stored into structures, such as a fruit, like in this example of strawberries. Or it could even be shuttled under the ground in the form of like a root, like a potato. But potato is not technically a root; it's a modified stem called a tuber. But if you had something like a carrot, that would be a storage of all this extra sugar for the future. So it depends on what you want. If you want to store sugar for the future, you might want to go polysaccharide. You could store it in these sweet structures. Now, simple sugars like monosaccharides and disaccharides are inherently more sweet in terms of taste. So this might be a strategy that plants employ in order to help seeds to disperse by creating fruits, which are delicious for animals to eat. And so here we have monosaccharides. So monosaccharides are a simple sugar. A disaccharide would be two sugars held together. Oligosaccharides, again, it's somewhat vague, but it means a few sugars. And then polysaccharides, you're literally dealing with hundreds to thousands of sugars. So all of them have saccharide like this. So it's mono, di, a few, and then many is what we're dealing with here. And so it'd be great if you could recall some examples of each of these and be able to discuss their function. That would be a good goal. And so let's start off with monosaccharides. They're the smallest of the carbohydrate, and they serve as a source of fuel. And again, ultimately, the animals and plants are using carbohydrates and burning them. And when I say burning them, I mean combust them for a source of energy. Usually the breakdown occurs in the cytoplasm in a process called glycolysis in terms of carbohydrate metabolism or catabolism, the breakdown of sugars. And it's often finished off in the mitochondria, so a fuel source. Simple sugars are burned for fuel. But these simple monosaccharides, if they're produced in numerous amounts, cells can also take apart these monosaccharides and use them as a source of carbon for other molecules that they need, for example, maybe amino acids. And ultimately, they could be joined together to form lipids if there's enough fuel at the moment. Lipids are another source of energy, and so carbohydrates can be converted into lipids. That's another conversation that we'll have later. So monosaccharides in general, just wanted to say a little bit about a carbohydrate. Do you notice the elements that make up a carbohydrate are often carbon, hydrogen, and oxygen? There is an example of a carbohydrate I'll introduce at the very end that contains nitrogen as well, but most of the sugars are CHO. And they're not only CHO, but they're in this ratio of 1 carbon to 2 hydrogens to 1. So it's kind of like a carbon water carbohydrate, a carbon water, CH2O. Now, depending on the number of carbons, you can get different kinds of monosaccharides. You could have, if this was 3 carbons, this would be 6 and then 3 oxygens. So it's basically 1 to 2 to 1 carbohydrate. Glucose is like the most popular, the favorite carbohydrate. And this has a molecular formula of C6H12O6, but you can see the 1 to 2 to 1 ratio there. Also, we like to name a lot of these sugars, not always, but we like to name them with the suffix "-ose" at the end of it, like for example, glucose. That's that suffix at the end, "-ose." We'll be seeing this a lot. Here's a linear structural molecule of glucose right here. You can see 1, 2, 3, 4, 5, 6. Here is a sort of a cartoon drawing of glucose as well. You can see the carbons forming a ring right here. And I'll talk a little bit about straight chain and ring in a moment. One thing I want to say about these monosaccharides is that a characteristic of a monosaccharide is that it has, here's the parent straight chain carbon, hydrocarbon, but it has many hydroxyl groups. Notice I'll just circle them here. See, it has a hydroxyl, 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 hydroxyl. So it's multiple hydroxyls. So what of that? Well, that means that it can readily form a lot of hydrogen bonds with water. And so carbohydrates, monosaccharides, are extremely hydrophilic. They adhere to water. So very, very polar carbohydrates. That's fairly important. They can be classified also by the fact that they contain a carbonyl group. A carbonyl group, remember, is the C double bond oxygen. And if it's at the end, like this, of the sugar, like this, it's considered to be an aldehyde. And the straight chain could form into a ring. And what's fascinating about that is in an aqueous solution, it tends to be more stable in this sort of ring formation. But you could still see the polyhydroxyls coming off right here and here like that. So the sugar being very polar. Now, I was talking about the carbonyl being at the very end. If it's at the very end like this, for example, glucose, it's an aldehyde. Do you remember this? An aldehyde is a functional group, C double bond oxygen. Or it's an aldose if it's an aldehyde sugar, aldose. Or if the sugar has a carbonyl group C double bond oxygen, and somewhere in the middle of the molecule, it's considered to be a ketone, or in this case, a sugar, ketose. So 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. What's interesting is this person over here, this molecule, it happens to be glucose, and this one happens to be fructose. And what's interesting is these are isomers of one another. Iso meaning the same molecular formula, different structures. They're structural isomers. And so the difference is sugar glucose is a aldose and sugar fructose is a ketose. And significance of this will come up later in terms of their sweetness and their functionality in the cell. Okay, and so we can also classify, I may have mentioned this, monosaccharides could be classified whether they have three carbons or 5 carbons, or in this case 6 carbons are shown here in green, 1, 2, 3, 4, 5, 6. And so you can have trioses for 3, you can have pentoses for 5, or hexoses. Glucose happens to be hectose, meaning 6 carbons. Okay, and then I mentioned that their sugars are most comfortable in a ring formation in an aqueous solution, and you can kind of see how that might happen here. There's no addition. Everything's accounted for. You can see how the aldose is located over here, and it comes together, and so you could abbreviate. Do you see how the carbons, in terms of the ring, it starts over here on the right. This is the carbon number 1 or prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime. You could abbreviate it like this. You can draw it like that. You have to include the oxygen. And so this is a very poor drawing. Let me do that again. So this represents the ring of sugar. And of course, let me highlight it with a different color. This would be carbon 1, 2, 3, 4, 5, and then 6 up here. 1, 2, 3, 4, 5, 6. You can see here 1, 2, 3, 4, 5, 6, and you can see the hydroxyl groups coming off of it right here. And as I said earlier, that monosaccharides are primarily fuel for cells. Cells burn monosaccharides for energy. So monosaccharides are converted to energy. And though that's kind of mysterious, I hope you're intrigued by that. We can talk about sugar metabolism in the future. It's a great conversation. As mentioned before, you could also break up a monosaccharide and use the carbons for constructing amino acids. Obviously, you'd have to add an amino group to it and change it around. And also, you can break up simple monosaccharides into fatty acids if you wanted to store the carbons in this molecule, fatty acids, which are components of triglycerides for extra energy. And so one of the things I wanted to point out is that these hydroxyl groups, whether they're in a down direction or an up direction, is fairly significant. In organic chemistry, we like to use the term alpha, which is the Greek letter A, or beta. In this case, it's either up or it's down, so it's either A or B conformation. So if it's in a down orientation, the hydroxyl group, it's in an alpha. So alpha hydroxyl is in a down. Beta hydroxyl would be in an up orientation. So glucose could either be alpha or beta glucose. And this is going to be important when you start linking these sugars together to form disaccharides, and even more so in polysaccharides. This alpha beta orientation will affect whether it's difficult to hydrolyze the polymer or not. In other words, break it down. So I want to point it out. Alpha down, beta up. Okay, so getting back to these trioses, which is 1, 2, 3 carbon, 5 carbons or pentose sugars, 1, 2, 3, 4, 5, or hexose, 1, 2, 3, 4, 5, 6. Do you notice it's always 1 to 2 to 1, 1 to 2 to 1, 1 to 2 to 1? And then you can also classify it across this way as being aldehyde sugars or ketose sugars. And so let me just name some of these monosaccharides. This one's a very significant one. All of them really are. Glyceraldehyde. So glyceraldehyde is a sugar. Ribose, you might recognize this somewhere from your science past. Ribose is the sugar found in ribonucleic acid. And then these hexose sugars are rather significant. These happen to be all isomers of one another: glucose, galactose, and fructose. Okay? So let's continue this. What do you get if you join two monosaccharides together, and if you answered disaccharide, you'd be correct. And again, in order for that to take place, it's a chemical reaction involving an enzyme to do this because it's energetically not as favorable. You would liberate water. You would remove that hydroxyl and that hydrogen, and that's a dehydration synthesis or a condensation. So if you connected glucose and glucose together, you'd form the disaccharide maltose. It's a new sugar. Maltose is a disaccharide. Notice there's two sugars. Now, what's up with the 1,4 glycosidic linkage? Well, the linkages between monosaccharides in a disaccharide or even in a polysaccharide are considered to be sweet side linkages or glycosidic linkages. Why is it called 1,4? Because if you remember coming around the horn here, this is 1, 2, 3, 4 connected to the 1, and there's an oxygen. It just looks a little weird because the oxygen's so much extended, but it's just an ordinary two bonds connecting the sugars together. So it's a 1,4 glycosidic linkage, and I might add an alpha 1,4 glycosidic linkage because it's in a downward orientation. Maltose is a really important sugar, and it's formed by two glucoses together. You might be familiar with maltose. It's somewhat famously used in the creation of beer, or maltose for malt liquor. And then another even more famous sugar of all. I'd go this far. I might not win this in a debate, but I could say subjectively the most famous sugar of all, like the most famous reindeer of all. I think you know who that is. But the most famous sugar of all is sucrose. It's so famous that people are willing to just simply call it out as sugar. Pass the sugar. We need some sugar. Can I add some sugar in this? The recipe calls for sugar. Sucrose is a disaccharide. It's not the only sugar. All of these things are sugars, but sucrose gets to be called sugar. I'm not really sure why that's the case, but sucrose is joined from glucose and fructose, and it's major in plants. And then this is also a famous disaccharide, lactose. You might know it. It's the sugar found in milk. And so what you have here is glucose and galactose joining together. And what's of significance here is that the glycosidic linkage is in a beta orientation. So it's a beta galactosidase linkage, which, as it turns out, is difficult to hydrolyze. It's difficult to hydrolyze, in other words, to break that bond. It's a lot easier to hydrolyze an alpha bond. And so let me just show you a simple, quick video on this. So if you had glucose and fructose, together and you joined them, water would be liberated, and there's your table sugar, sucrose, that people like to just call sugar. So that's an alpha linkage right there. And then if you had galactose and glucose, you would form the disaccharide lactose. Notice it's in a beta. This is milk sugar. And then if you had glucose together, it would form maltose sugar. And the thing about maltose is that if you had starch, and I'll get to this in a moment, but if it's starch, which is a big polysaccharide of glucose, ultimately when that's broken down, it's first broken down into little maltoses, and then maltoses are broken down further. And then there's enzymes that help speed up the hydrolysis. And so you can call these enzymes out, like the enzyme that speeds up the hydrolysis of sucrose is sucrase. The one that speeds up lactose is lactase, and then maltase. So you just take the suffix away. And so let's come back here if we can. Maybe. Maybe not. Maybe yes. Okay. So there's lactose. And here's the creation of sucrose, table sugar. It happens to be a 1,2 alpha. 1,2 alpha. Here's glucose, here's fructose. Do you notice how fructose is shaped a little bit different than glucose? This is an aldose, this is a ketose. And so here's sucrose. So this is a dehydration synthesis of sucrose. If you wanted to break it down, you would add water, and then it would split it back up into glucose and fructose. Now, just a little break a little bit from the biochemistry of this in terms of practicality. What does it matter whether or not you're dealing with fructose or sucrose or glucose or maltose, etc.? Well, it's fairly significant if you work in the industry that uses these monosaccharides and disaccharides as sweeteners. It's significant because there's literally, I'd go as far as to say billions of dollars at stake, whether or not the recipe for your soda has fructose in it, or whether it has glucose in it, or galactose in it. It could make a difference. And so arbitrarily, people were assigned different drinks of sugars, and they were asked to rate it on a scale, determining what is the most sweet, what is the least sweet. And so apparently people like sweet things more than less sweet things. And so fructose jumps out as being the winner in terms of most sweet. And so there's certain companies, and you could investigate this, although these are trademarked recipes, that I don't know that these soda companies would totally reveal it. But I believe this is not necessarily factual, but I believe Pepsi uses fructose, and I believe Coca-Cola uses sucrose in terms of its recipe. And so it doesn't, just because it's more sweet doesn't mean everyone loves it. Like, you could love that or love this, or not love it at all. But polysaccharides are monosaccharides linked together to form many chains. And so why would you do this? Why would you make many sugars together? Well, two very important reasons. To store a tremendous amount of energy. Can you see how there would be so much more energy in a polysaccharide than in a monosaccharide, just one? So you can get a lot of energy storage this way, and it can also be structural. So plants in particular use polysaccharides to construct their walls, which were really important to help maintain their shape and the integrity of the cell. And so wood is made up of polysaccharides, and the polysaccharide happens to be cellulose. And so polysaccharides are formed by polymers, so we can say polysaccharide, and there are hundreds to thousands of these monomers connected together with the glycosidic linkages. And again, energy. Very important storage of energy, polysaccharides. And then as you need it, you just simply hydrolyze and pinch off a sugar as you need it. And so it's great that way. It's sort of like a big chain of beads, and you just start cleaving the beads every time you need a little bit of energy. And they're also great building blocks, as I was mentioning, for organisms as well. And so one of the most famous polysaccharides of all is starch. And so we equate it with things like flour. Plants produce it, for example, wheat makes it, and then we crush it down in the mill and turn it into flour, and then we can use that flour, which is starch, as a source of all kinds of foods that we like to eat, like, for example, bread and pastas and things like that. And so starch is a complex polysaccharide or complex carbohydrates. You may have heard of that before. So here are the chains of glucose that are held together. Starch happens to be made up entirely of glucose. So big chains of glucose like this. So each one is here. So this is a monosaccharide, and this is a polysaccharide. And it's a great storage of energy. Now, starch is the common name. The widely accepted name for starch is actually amylose, or amylose, right there. It's a big chain of glucose. Or if it's a little bit branching, coming off to the side, it's called amylopectin. So both are storages of glucose. This is what we can say this. And now, where do we store this? We could store a little bit of starch. As you can see here, this is the transmission electron micrograph. Yeah, this of a chloroplast, which is usually green. Plants make the glucose in the chloroplast and then store a little bit in the form of starch. But the truth is that chloroplasts aren't the best place to store the starch. There's some clear plastids that are called leucoplasts that are found in roots that store a lot more starch. And so potato, if you think, is a tuber, a modified stem that grows under the ground. Plants really love to store their glucose in the form of these potatoes. And so these potatoes, like when we eat a potato for eating French fries or for eating potato chips or baked potatoes or anything potato, we're getting a good source of energy because we're eating starch. And so that's excellent. And so what's fascinating is that animals also store their extra sugar in the form of a polysaccharide called glycogen, sometimes commonly known as animal starch. Now, what's interesting is when we consume glucose in our diet, like for example, if we eat a lot of pasta, we could digest that or hydrolyze that into simple sugars, but then we can link it back up again into a polysaccharide called glycogen. It's very similar in structure to starch. And then we could store that in our muscle. It's a great place to store glycogen because ultimately this is where it's going to be consumed. We could store it in our liver and in our muscle because back over here you can see inside of a cell the glycogen would then be broken down into glucose, and then glucose would ultimately be broken down in the cytoplasm and in the mitochondria for energy. And so we have about a day's storage of glycogen in our muscle and our liver. We don't want to store all of our extra energy in the form of a polysaccharide because it's so hydrophilic that water would cling on it and it would make us incredibly heavy, because you may know that muscle's pretty heavy, and so it has a lot of glycogen in it, and so therefore it also has a lot of protein, which is also hydrophilic, but it would make us so heavy that we wouldn't be mobile. And so I just wanted to point that out. Again, I'm distinguish between starch and glycogen and the next polysaccharide of significance, which is cellulose. And one of the big differences is this alpha and beta orientation in the glucose. Remember, if it's in a down orientation, it's alpha, and if it's in an up, it's beta. Okay? So check this out. If you have starch, what's holding all of the glucoses together are the alpha 1,4 glycosidic linkages. That's starch. Obviously, it's not all the sugars are being shown. It's just 4 of them. Now, check this out. There is a very important polysaccharide out there called cellulose. And cellulose is where plants use this to make up their tough cell walls. And the reason that it's a great building material ultimately is that it's difficult to break down. You don't want to be building something strong with a molecule that's weak. And so what's interesting and different about starch and cellulose is that every other sugar, there's a beta orientation. So this is beta alpha, beta alpha, beta alpha. So cellulose is a major, this is important, component of plant cell walls, and it's because of the beta orientation right here, the up. And so it makes it very difficult to break down, and so it's very strong. Cellulose comes from plant cell walls, as you can see here. Now, inside the cell wall is the thin phospholipid bilayer membrane right in here. And I find this to be kind of a great picture because these little green guys inside here are the chloroplasts. And then you're like, well, why are you talking about that? Because that's where sugars are made in the chloroplasts through photosynthesis. So each one of these glucoses are produced in here, and then ultimately they're linked together to form the cell wall. And so there's lots. Now notice here, it's beta alpha, beta alpha, beta alpha. So this is cellulose. It's a very important structural polysaccharide which helps plants survive. And so ultimately, these polymers are very strong and they form these microfibers. And these are the basic building blocks of plants right there. And then ultimately, when you think about it, it's not only the cell walls of leaves, but you're talking about the cell walls of vascular tissue as well, which are found in the trunk or stem of plants. So here's a big chain of cellulose, and then ultimately, this is scanning electron micrograph. The cell walls right here, this is of vascular tissue called xylem. Ultimately, these big vessel elements and these tracheids are made up of cell walls, and sometimes they even have two cell walls, a primary and a secondary one. And this is this rigid polysaccharide material. And this is where water is able to travel up a plant like in a straw in this tissue. It's pretty interesting. And then the water adheres to the side of the cell, and then through cohesion, capillary action, both of them together, are pulled out from the top of the plant. And that's how water moves up the plant. Now the thing is, if cellulose is so strong and it can't be broken down easily by animals, then what's the point? It doesn't provide any energy. But we don't necessarily eat it because it has a lot of energy. We would eat starch because it has a lot of energy. If something cannot be broken down, think about this. If it can't be broken down from a polymer, it's going to remain a polymer, and cells aren't going to be able to absorb it because it's too huge. And so therefore, it provides like fiber. So it's an insoluble fiber is what is actually secreted in the form of feces. And so most of what feces is undigested cellulose material. So we eat fruits and vegetables as a source of not only sugar for energy, but the cell walls provide a lot of fiber. So it's double goodness. We get some energy, but we also get indigestible fiber, which is the cellulose, which helps to move the food through the digestive system. And so it's very helpful that way. And so, for animals that make a living from eating a lot of cellulose, there's not a lot of starch in grass and that sort of thing. They have a digestive tract that has a lot of microorganisms that are living in symbiosis with them. And it's the microorganisms, which are the ones that are digesting the cellulose. There are prokaryotes that can break down cellulose. Likewise, animals like termites, which do a lot of eating of wood, which is a lot of cellulose, can use these microorganisms to break down cellulose into glucose, which is really important. And then finally, there's another interesting polysaccharide that I wanted to mention that's kind of significant. It's kind of like cellulose. Do you notice here it's like glucose, beta, and then alpha, like that? But what's interesting is that it has this little nitrogen functional group attached to it, to the sugars. And so this polysaccharide is called chitin. And it's also a structural polysaccharide, and it's found in the exoskeletons. That means outside skeleton. We have an internal skeleton. The outside skeleton of the phylum arthropods. They're arthropoda, so jointed feet. So insects and spiders and crustaceans, it happens to be the largest phyla. So all of these shells from these organisms are made up of this. In other words, the shell of a crab is made up of chitin, and it's a polysaccharide with these nitrogen groups on it. But what's interesting is what gives it a little bit of its rigidity. It would be a little bit more leathery if it didn't have calcium, and so there's the addition of calcium carbonate to that chitin helps to keep it a little bit strong and protective. And also fungi, in their root system, they also contain chitin in their mycelium. And so that's kind of important too when you consider the fact that fungi is so numerous. And so that's an important polysaccharide. So chitin is significant. And cellulose. One might say that cellulose is the most abundant carbohydrate in the world, polysaccharide that is. because it's found so prominently. And so I hope you enjoyed this conversation about carbohydrates, both simple ones and complex carbohydrates. Thanks for watching.